All right, ladies and gentlemen, we are going to talk about sexual reproduction, specifically the process by which we get gametes, which are the sex cells of an organism, so sperm and eggs. I'm trying some new technology, so bear with me if there are problems. So, we're going to be going over this. It's very similar in process to mitosis, and I will try and put this into some evolution context, just because it might help explain a few of the strange things about meiosis. I will admit to you, when I was in high school myself, I was very confused by meiosis for a few reasons. So, originally, all organisms on the planet, back when they were bacteria, were all asexual. They're kind of like haploid organisms, meaning that they only have one copy of each gene. And then they reproduce by a process called binary fission. Binary fission does not produce genetic variability, so it produces nothing but, that's not, they produce nothing but clones. And while, that is fine when you have environmental changes, though, that can mean that your population is vulnerable. Without genetic diversity, your population can be killed by a single event or a single disease. So there are two ways to increase genetic variation in a population. One is to increase the mutation rate, but that is a kind of dangerous proposition because you have the risk of having harmful mutations, and that chance of developing harmful mutations is a lot stronger than the chance of developing any beneficial ones. So that's not really a good solution for organisms. The second is to increase genetic recombination. So combine different combinations of genes and hope that one of them is going to be a winning combination that's going to survive. So one of the things that we're going to focus on is we're going to focus on where genetic recombination, where sources of genetic variation occur throughout this whole process. Right? So, and you may think to yourself, okay, well, bacteria only have a single chromosome. Why don't they have more than one chromosome? Well, it was, they were the first organisms to evolve, and generally the first things to evolve tend to be simpler. However, it became beneficial to have more than one copy of your chromosome. And the reason is, if you had a mutation in an essential gene, but you are only containing one copy of that gene, that means any mutation could be fatal. However, if you've got an extra spare backup copy of that gene that's unmutated, then that could be enough to allow you to survive, while also allowing that mutated gene to change your chance of survival or change the chance of your offspring's survival somewhere on down through the generations. Usually the original version of a gene is called a wild type allele. And we're going to revisit that name and that terminology and all of that when we talk about genetics in a couple of weeks. So, duplicates of chromosomes are called homologous pairs, and that's going to be an important term to keep in mind. And so all eukaryotic organisms are at least some point in their lifespan diploid. And that means that they have more than one copy of each chromosome. So we can talk about sets of chromosomes. A set of chromosomes is each individual unique chromosome in an organism's genome. Most eukaryotes, like I said previously, have both diploid and haploid parts of their life cycle. Diploid means that you have two copies of every chromosome. Haploid means you only have one copy. And if that's confusing, don't worry, we're going to go over it in more detail. You also have some organisms that are polyploid. Polyploid organisms have more than two sets of chromosomes. It's rare to have this happen in animals, but it's very common to happen in plants. And in fact, most of the plants that we grow commercially for food tend to be polyploid. excuse me, because when they have more than two copies of every gene or of every chromosome, it tends to result in larger fruit. So we tend to breed for polyploidy. And then when we're talking about how many chromosomes we have in a set, we call that number n. So n can vary depending on what organism you're looking at. So if you're looking at, a plant, n could be a large number. The n for humans is 23. That means every set of chromosomes we have has 23 chromosomes in it. Okay, moving on. So let's talk about what I call ploidy. Haploid cells have only one copy of each chromosome. So in this example right here, n equals 3. So a haploid organism would have one N or 3 chromosomes, and a diploid organism would have twice that many because it has chromosomal pairs. So those are homologous chromosomes, which we usually call maternal and paternal, meaning from the mother and from the father. Diploid organisms of this type would have 2 times N or 6 chromosomes. Some other terminology that is important to know. The sex cells in an organism are gametes. Those are sperm and eggs. Those are the haploid cells. And then diploid cells are usually somatic or body cells. So now that we've introduced the term homologous chromosomes, let's go ahead and talk about what these are. These are chromosomes that code for the same genes or the same traits, and they will generally have those on their chromosome in the same location. So you can map out a chromosome and you can say, okay, gene A is up here, gene B is over here, gene C is here, gene D is here. And when you look at homologous chromosomes, they'll have those genes in the same location. So a genetic locus is where a gene is located on a particular chromosome. So, at a specific locus on a chromosome, you'll have a specific gene, and at the same locus on a homologous chromosome, you will have a gene for the same trait, generally for the same protein. So usually one of each pair of homologous chromosomes is your maternal copy, meaning it's from your mother, and the other is your paternal copy. Usually it's meaning it's from your father. Generally, there's not a lot of, while there is some modification to DNA that can occur based on whether it's from your mother or your father, in general, with the exception of the sex chromosomes, if you're a male, it's kind of hard to look at your chromosomes and say, okay, you got that one from mom, you got that one from dad. And we're going to talk about one or two of the reasons that make that a difficult task in a little bit. So, before we have any sort of meiosis is a type of cell division, and it is divvying up DNA. One of the things I always found really confusing when I was in high school biology, I didn't understand why you would replicate chromosomes before meiosis. I was like, that seems like a lot of extra work, wasted of energy when you can just split apart your homologous chromosomes. But um, meiosis is actually kind of a process that evolved from modifications to mitosis. And so it's going to have a lot of the same structures, it's going to have a lot of the same phases, and it starts out the same way. So since mitosis started out with replicating DNA, replicating all your chromosomes, meiosis has that as well. And this is going to highlight something that we'll see in evolution multiple times, and that is that evolution does not always lead to the most efficient systems. So let's look at how many sets of chromosomes are found in a cell after S phase, after DNA replicates. So this represents a pair of duplicated homologous chromosomes. So each of those X shapes contains 2 identical copies of a chromosome. So you have 1 set of chromosomes there, 2 sets of chromosomes there, 3 sets there, and 4 sets there. So this is important to keep in mind because when we look later at the results of meiosis, what we're going to see is that we're going to produce 4 different cells, each of which is going to contain one set of chromosomes. And the reason why we have 4 cells is because we have 4 sets of chromosomes in the cell before meiosis occurs. So, before meiosis can happen, you duplicate your chromosomes just like in mitosis, but a different step happens, and that is that homologous chromosomes pair up with each other. Then you go through 2 divisions, and that is going to divide your cytoplasm twice. But more importantly, it's going to divide the number of chromosomes twice, so that where you may start with, say, 2N chromosomes, that's going to take you down to 1N chromosomes. So we call it a reduction division because it's reducing the number of chromosomes. So meiosis 1 is where you separate homologous chromosomes. It's going to make 2 cells, each of which is going to be 1N. So this is we're going to talk about specifically in humans. That's why everything's in multiples of 23. So in humans, after the end of meiosis 1, you're going to have 2 cells formed with 23 pairs of sister chromatids. This is basically where you do the reduction. You go from 2N down to 1N. At the beginning of meiosis 2, that's when you separate your sister chromatids that you produced from DNA replication. And at the end of that, you're going to have 4 cells, each of which is going to have 23 single chromosomes. You start haploid, and you're going to end haploid. So there's no difference there. So we're going to go through each of these stages. So just like mitosis has prophase, metaphase, anaphase, and telophase, meiosis has the same things. There are a few modifications here and there. For example, during the first meiosis, during prophase 1, you have similar steps happen. You have a protein spindle form between 2 pairs of centrioles. You have your nucleus break down into bits and pieces, so that way your chromosomes have room to move. But what's different is that you form these pairs of homologous chromosomes, and that's going to become important in a few moments. It's also going to be a source of genetic variability. So you pair up your homologous chromosomes, so that way you can line them up in the middle of the cell. And this is metaphase 1. So notice you have pairs of chromosomes lined up at the center of the cell. Each of these pairs is going to be connected to one of the protein spindle fibers, and during anaphase 1, each homologous chromosome is going to be separated from its pair. Now notice we still have sister chromatids here, so that's why we still have those X shapes for our chromosomes instead of just having a single line. And then telophase 1 can also be accompanied by cytokinesis, although in some cells when that's happening, cytokinesis doesn't happen until the very end. Anyway, so an important step that happens during prophase 1 is when the homologous chromosomes pair up, then you have an event that can happen called crossing over. So crossing over happens at these spots called chiasmata. Literally means an X. It's basically a crossing point, and this happens at like homologous locations, so at the same genes. So since homologous chromosomes have, the same genes in the same order, you can have gene A right here, where you have a dominant copy, and you can have the same gene over here, where you have a recessive copy. Well, let's say you had gene B up here, dominant copy, and gene B up here, recessive copy. Well, originally, the only combinations you can have are all dominant or all recessive. However, after crossing over, you have what are called recombinant chromosomes. This one would be dominant B, but it would be recessive A. So now you have different combinations you can make. And this one would be recessive B and dominant A. So this is a source of genetic variation. This is recombination, basically combining again genes, and it produces more variety in offspring so that there's a greater chance that one of those offspring will survive and pass on your genes. Um, this is also a point at which it's important to note that your chromosomes don't have to separate based on their source. So maternal and paternal chromosomes, like I said, don't have any markings in them saying that, oh, okay, this one came from mom, this one came from dad. So when they line up at the metaphase plate during metaphase 1, they don't necessarily have to end up on the same side. So you have a couple of possibilities if we look at two pairs of chromosomes or two sets of homologous chromosomes. Here we see a possibility where the maternal chromosomes line up on one side and the paternal chromosomes line up on the other side. So you don't have any different combinations of genes. You still have capital A, capital B, capital A, capital B, or lowercase a, lowercase b, meaning recessive. However, in possibility 2, you could get one copy of the paternal and one copy of the maternal in each of these chromosomes. And so then you can get different combinations. So now, instead of having all dominant or all recessive, you can have a combination of dominants and recessives. Now, keep in mind, this is called the law of independent assortment. Basically, traits sort independently of each other. That was what Gregor Mendel found when he was doing all of his genetics experiments. But what we know now, he didn't know about chromosomes. He didn't know that DNA was the genetic material. Now that we know this, we know that traits that are on different chromosomes sort independently. So that's going to be something important to keep in mind. So then prophase 2 happens. A new protein spindle forms. You generally don't reform the nucleus in between, so there's no need to fragment it up again. Then during metaphase 2, now you have your pairs of sister chromatids lined up at the center. During anaphase 2, they're going to separate from each other. Notice, by the way, that we have these recombinant chromosomes here that are basically kind of Frankenstein copies with part maternal, part paternal chromosomes. And then finally, at the end, you have telophase 2. At this point, you do reform your nuclear envelope because these are going to become separate cells. And then you have a separate second cytokinesis, and you finally formed 4 different sex cells. So which sex cells are formed and all of that, of course, depends on the gender of the individual. Males make sperm through a process called spermatogenesis. Females make eggs through a process called oogenesis. There's some differences between them, though. So notice that in spermatogenesis, you have one original cell from which you actually make 4 different sperm cells. However, this process is different in oogenesis because first you make, you have your first division happen. And notice that this, what's called a polar body, is going to basically be released with a set of chromosomes, but it is not going to contain a lot of the cytoplasm. And this first polar body disappears. It basically dies. So it's just a way to kind of spit out some of the DNA. Then before fertilization can happen, you have a second polar body that's just kind of another place to throw away, to also throw away the extra genetic information. And then finally, you have fertilization happen where a sperm and an egg combine, and that will make a zygote or a fertilized egg cell. The reason why this is different is because when a sperm fertilizes an egg cell, the main contribution a sperm makes is going to be its DNA. A sperm cell is mostly genetic material packaged with some mitochondria to help kind of give it energy and a flagellum. And its job is to get that DNA to the egg cell before any other sperm cell does. Each sperm cell competes with the other. So even in that case, it's kind of a survival of the fittest. But once it reaches fertilization, the egg cell is going to be the source of most of the organelles. So most, if not all, of the mitochondria that you get come from your mother. All of the cytoplasm that you got came from an egg cell in your mother. So since it needs to be self-sufficient and it needs to be able to survive with whatever resources it has until an egg cell can implant in the uterus, it needs to contain most of the cytoplasm. That's why you basically have these kind of throwaway polar bodies that are formed. They're just ways to spit out the extra DNA. So sometimes errors can happen in meiosis. A common one is called non-disjunction. So a junction is a joining between two things. Disjunction would be separating them, and non-disjunction is a failure to separate. It's a super complicated compound word that's basically saying, hey, two things that were supposed to come apart didn't. So this can happen in two different spots. It can happen either in meiosis 1, where a pair of homologous chromosomes don't separate, or it can happen in meiosis 2, where a pair of sister chromatids don't separate. The end result, though, is that you get these gametes that have uneven numbers of chromosomes. So say that n equals 2 for this species. Sorry. Notice that with the non-disjunction in meiosis 1, you end up with 2 of your gametes that have an extra chromosome and 2 of your gametes that are missing a chromosome. With the meiosis 2 non-disjunction, you have 2 normal gametes with the proper number of chromosomes. You have one that has one less chromosome than it's supposed to, and then you have one that has an extra chromosome. What can happen is if this gamete fertilizes or gets fertilized by a normal gamete, it can result in something called a trisomy. A trisomy is when you have 3 copies of a chromosome instead of the normal 2. Now, in most cases, this would be fatal. However, with some of the very smallest chromosomes, there are survivable trisomies. For example, the most common trisomy is Down syndrome. Down syndrome is trisomy of chromosome 21. Chromosome 21 is the smallest non-sex chromosome that exists, and depending on the severity of the chromosomal duplication, you can have either mild impacts on a person's development, or you can have very severe ones. So there are delays in physical growth. There are some characteristic facial features that all children with Down syndrome have, and they may have an intellectual disability as well. And like I said, the severity of the disability and the other physical impacts will be due to how much chromosome got duplicated. There's also a couple of trisomies, or in one case, not a trisomy, but still a result of non-disjunction that can result with the sex hormone. So Turner syndrome, you only have one sex chromosome, that's a single X chromosome, and these people are phenotypically female, so they develop as female. However, they may have some slight physical differences from double X females. However, it's probably not noticeable until about puberty, because they only will start developing a menstrual cycle and secondary sex characteristics if they are given hormone treatment. Klinefelter's is another case of a trisomy. Instead, in this case, it's an extra chromosome, an extra X chromosome. But while it's an extra 2 X chromosomes, one of those there's one of Y chromosome in there, which will cause a phenotypically male phenotype. However, with that extra X chromosome, it can cause some small amount of feminization, like smaller genitals, a little bit more breast growth than in, genetically XY males, and can result in lower libido as well. So mitosis and meiosis are very similar. They share the commonality of the phases, both in the names and also what happens between them, but they have very different outcomes. So mitosis produces clones. So if you start with a somatic or body cell that's diploid, that's what you're going to produce at the end of mitosis. So these are producing just exact copies of a cell. Meiosis, on the other hand, changes the genetic composition of the cell that's produced. Instead of having a diploid organism, you're going to randomly have half of the chromosomes in one gamete, half of the chromosomes in another gamete. And that is also a difference. It produces gametes, which are sex cells. So we're not going to pay much attention here, but I did want to point out that regardless of what eukaryotic organism you are, you still have haploid and diploid stages in your life cycle, and it all depends on fertilization and then meiosis. So an animal has a life cycle that we should hopefully be familiar with. Meiosis produces sperm and egg cells. Those sperm and egg cells are haploid. They are, not a separate stage of development. They're just cells. They combine to form, during fertilization, to form a new organism, and that new organism for the duration of its lifespan will be diploid, although once it reaches sexual maturity, it will produce haploid gametes. So, meiosis and fertilization are key here. Plants also have haploid and diploid parts of their life cycle. Depending on the specific type of plant, you may have a completely separate organism that is haploid for the period of its life, but you can also have other organisms like angiosperms, meaning flowering plants, that actually don't have a separate haploid stage of their life cycle. But notice that they have in common, they have meiosis and fertilization. And then, if we look at our least examined, at least in this class, member of domain Eukarya, fungi, they actually have what looks like the opposite life cycle from us. Most of their lifespan is spent as a haploid organism. And then they produce gametes, which then fertilize to produce a diploid form, which is usually not an independent life form, which will then undergo meiosis. And meiosis is going to produce spores. Spores are, the reproductive structures generally of fungi, and there's also the things that you can be allergic to, and they will go off and they will land somewhere and grow a brand new organism. But once again, we see the trend: meiosis and fertilization are key. All right, hopefully that worked out, and you'll get your next video notes soon.